So for variant analysis, you typically use BWA-MEM. Why BWA-MEM? Well, MEM because it can handle longer reads, and for variant analysis, you're typically going to work with longer reads. And why BWA? Well, because BWA is considered to be a bit more accurate than bowtie. It's not a big difference, but generally people agree that BWA is a bit more accurate. Another reason why many people go for BWA-MEM is that when you are mapping to the human genome, and again, for variant analysis, most data come from human, BWA-MEM allows you to map against the full genome. And what do I mean with the full genome? Well, not only the primary assembly. I told you earlier, if you're going to download a reference genome, in most applications, you're going to go for what is called the primary assembly. What is the primary assembly? It's the non-redundant haploid genome. But on top of this primary assembly, there are extra sequences, and they are called alternate contigs. And they contain the sequences of large variations of sequences in the primary assembly. And these variations are called alternate haplotypes. Now, what is the issue here about these alternate haplotypes? Why don't you usually use them for mapping? Well, because these alternate haplotypes, they contain a part that is different from the original sequence in the primary assembly, but they also contain a part that is the same as the original sequence in the primary assembly. So when you have reads that map to the part that is the same, these reads will be considered multi-mappers because they will map to a sequence in the primary assembly, and they will also map to sequences in these alternate contigs. So that's multiple mapping locations, so it's a multi-mapper. All applications ignore multi-mappers in the next steps after the mapping. So you lose all these reads. And this is why generally people map to the primary assembly. Then you don't have this issue. Now, for variant analysis, it can be very useful to have these alternate contigs. You can imagine that that's maybe one of the things that you want to look at. So this is why a version of BWA-MEM was developed called BWA-KIT that allows to map against these alternate contigs. How does it work? Well, it will first map the reads on the primary assembly, and then the reads that do not map well on the primary assembly will be mapped against the alternate contigs, but they will not be flagged as multi-mappers. So, and the combination of the BWA-MEM mapper with the version of the human genome that includes these alternate contigs is called BWA-KIT. So on the links on the slide, you can find more information on it. At this moment, it's not yet included in the Belgian Galaxy, also not in the other Galaxy servers, so also not in EU or in the American Galaxy server, but it is in the process of being built in the Belgian Galaxy. So it's there, but we still have to check it, so it's not yet public. So these alternate contigs, what are they? So they are just extra sequences, large sequences, contigs, extra sequences that contain a variety of things. So some of these alternate contigs contain these complex variants, as I explained, so variations on sequences in the primary assembly. And many of these variations are linked to the immune system. HLA loci, for instance, they are an example of sequences, alternate haplotypes that are in these alternate contigs. But they also contain other things like fixes of errors in the primary assembly, or fixes of gaps in the primary assembly, are variations that have been discovered and that are not yet in the primary assembly. So all these extra things are typically stored in these alternate contigs. And then when the next version of the genome is released, many of these sequences that are now alternate contigs will be included in the primary assembly. So all the error fixes and the gap fixes will be included in the primary assembly.